Hello everyone, I'm Esra Büşra and today I will be talking about my project named Computational Characterization of Ligand, Specificity and Promiscuity of Staphylococcus aureus NorA Efflux pump for ISMB 2022 and 3D SIG COSY. And my presentation will go like this. I will briefly introduce the project and I will talk about our aim, methodology and results, and then we will hopefully conclude. So bacteria are everywhere, as you know, and they cause 10% of the emerging infections. And we treat bacteria by antibiotics, but excessive use of these antibiotics can cause antibiotic resistance. And one of the mechanisms that bacteria use for causing antibiotic resistance is by mutations in efflux pumps. And Staphylococcus is a bacteria which is multiple drug resistant, and it can cause toxic shock syndrome, septicemia, etc. And NorA is a flux pump of Staphylococcus aureus. It is found in the transmembrane of Staphylococcus aureus, and it belongs to major facilitator superfamily. It is an antiport transporter, and it contributes to bacterial antibiotic resistance against antiseptics, biocides, and many others. And its structure has not been discovered experimentally, but we know that it has 388 amino acids and 12 transmembrane segments. And in our project, our aim was to characterize this ligand specificity and promiscuity of NorA efflux pump by computational methods. And for this purpose, we have obtained the 3D structure of NorA by homology modeling software. Then we have used molecular dynamics simulations to check the stability of this homology model. Then we have analyzed the binding pockets, and from molecular dynamics simulations we obtain different conformations of the protein. Then we have analyzed the binding pockets for each conformation. Then we collected the known inhibitors of NorA, and we have used molecular docking softwares to dock these known inhibitors to different binding pockets of different conformations. Then we analyze active binding residues, the amino acids that are forming the bonds with the inhibitors. Then we clustered these inhibitors based on binding sites and tonometric similarity scores. And this is the homology model of NorA. We have used AlphaFold generated structure of NorA as our template, and then we created this homology model by using this model. And then we, for molecular dynamics, we needed to prepare the protein, and because it is a transmembrane and it is inside the phospholipid bilayer normally. For that, we have used Charm GUI, which creates this environment for your simulations. Then we have used GrowMax for molecular dynamics simulations, and we have obtained a simulation which is 100 nanoseconds. And here you can see the RMSD plot, which shows the scores, the different conformations, the scores based on different conformations. And based on our root mean deviation score, our dynamic simulation was trustable enough. After that, we analyzed the different pockets by using dockside scorer, and we had obtained around 40 different conformations, and for each of them we have analyzed the binding pockets, and we had 178 different inhibitors. By using Autodock Vina, we have obtained different docking runs, and by using Flip, we have analyzed the binding residues that were forming the bonds between inhibitors and the NorA efflux. And let's go through the results briefly. And when we check the binding residues of NorA efflux pump, you can see that some of the amino acids and some of the locations were found repeatedly. For example, phenylalanine at position 47, isoleucine at position 19. These amino acids were found to be forming bonds with most of the inhibitors. And when we highlighted these regions, these amino acids, we have realized that they are really close to each other. So this site can be proposed as a binding pocket of NorA efflux pump. pump. And then we have analyzed the inhibitors based on their binding characteristics, the amino acids that they are binding. So according to that, we have found 3 distinct groups that are kind of forming bonds. And we have chosen one representative from each group. We have visualized them. Then we have done the same analysis for ciprofloxacin, which is a known substrate of NorA. Then we realized that ciprofloxacin shows similar characteristics with second group. And these groups, they are really showing different characteristics while binding NorA efflux pump, which is kind of explaining the promiscuity of NorA efflux pump. And then we have clustered the inhibitors based on terminator similarity scores. However, and we have found some inhibitors that were closely related with each other. However, when we compare this with the binding side characteristics of inhibitors, we couldn't find any significant results. So as a conclusion, we have obtained the homology model of NorA, and we have found the possible binding pockets and possible binding residues of NorA efflux pump. And with this approach, we have enlightened the promiscuous structure of NorA in silico by finding different groups that have different binding characteristics. And thank you all for listening. And if you have any questions, you can email me anytime. And hope you have a great day. Thank you.